Welcome back. So, in today's lecture, we are going to look at some quantitative methods to understand the drug receptor interactions. So, nature of the drug receptor interactions can be understood by trying to figure out what the stability of the drug receptor complex looks like. Okay. So, the more stable for the drug receptor complex is, the more difficult it is for it to dissociate. So, in order to understand this, we need to define a term known as dissociation constant. Okay. So, the assumption that we will be making is that the drug is in equilibrium with the receptor. What it means is that the drug interacts with the receptor and forms a receptor complex, drug receptor complex, and then it again dissociates to give you the drug and the receptor again. So, this, much like a chemical equilibrium that we are used to, can be defined by an equilibrium constant, which is K d. So, K d is nothing but the concentration of the drug multiplied by the concentration of the receptor divided by the concentration of the receptor complex. Much like an equilibrium A plus B going to C, the equilibrium constant is defined as concentration of C divided by concentration of A times concentration of B. Okay. Now, if you look at the reverse of this process is what you would get for the drug receptor complex. Okay. Now, the smaller the dissociation constant K d, that means that the drug multiplied by the receptor concentration is much larger compared to the drug receptor complex concentration. That means, this value is low and this value is high. Okay. Then when you divide these two, you would get a small number. Okay. That means that the drug receptor complex is extremely large in concentration, which also means that the drug has very high affinity towards the receptor. So, the median dissociation constant for inhibitor drugs in the market is about 20 nanomolar. Okay. So, what is a nanomolar? Nanomolar is, so if I consider molar and then if I divide this by 1000, I get millimolar, which again I divide by 1000, I get micromolar, and then finally, if I divide that by 1000, I get nanomolar. So, nanomolar is actually an extremely small number, and therefore, the dissociation constant for these drugs is quite low. Now, let us look at some of the factors that determine the affinity of the drug to the receptor. And before that, let us define the since it is an equilibrium constant, K equilibrium, as we have studied in fundamental thermodynamics, the equilibrium constant is related to the free energy by the following equation: delta G naught equals minus R T ln K. So, the spontaneous formation of a bond between atoms occurs with a decrease in the free energy. Okay. And similarly, when a drug binds to a receptor, there is a non-covalent bond that will occur, which also must have a negative free energy term. And we already know that delta G is nothing but a sum of the enthalpic term and the entropic term. That is, delta G equals delta H minus T delta S. So, we will not be going into too many details about the enthalpic component and the entropic component, but you can break it down into and try to understand that all of these factors are going to contribute to how well the non-covalent interactions are happening to make the drug bind to the receptor. So, the affinity of a drug for a receptor is a measure of how strongly the drug binds to the receptor. Okay. We shall define another term known as efficacy. Okay. Efficacy is a measure of the maximum biological effect that a drug can produce as a result of receptor binding. So, we have looked at in detail in the past several lectures that once the ligand binds to the receptor, it results in a conformational change which can activate a cascade of processes which results to signaling. So, efficacy is how well this biological effect occurs, whereas affinity is how well a drug binds to a receptor. Okay. So, it is important to understand the differences between these two. Just because a drug has high affinity towards a receptor, it does not mean that it is going to have high efficacy. So, for example, an antagonist which can bind with high affinity, but it can have no efficacy, that means that it may have no effect on the signaling cascade. Lastly, we will look at what is known as potency. Potency of a drug refers to the amount of the drug that is required to achieve a defined biological effect. Okay. So, let us say the defined biological effect is inhibition of proliferation, that is how to slow down growth of cancer, or decrease in pain, or increase in healing, or any process that we would define as a biological effect. So, the potency refers to this, and the smaller the dose required, the more potent the drug. Okay. Of course, you will appreciate that it is possible for a drug to be potent, but have very low efficacy. Okay. Affinity of a drug to a receptor can be measured by using a process called as radioligand labeling. Okay. So, what we do in this process is we take a ligand and we label it with a radioactive isotope, and since the radioactive isotopes are easy to detect, it would be easy for us to understand how well the radioligand is bound to a receptor. So, if we take an antagonist or a ligand and take one of the atoms in the antagonist or the ligand and label it with a radioactive isotope, now when you add this to a cell in culture or tissue, now what would happen is that it would go and bind to these cells or tissues, or receptors on these cells or tissues, and now you wait for some time until an equilibrium has been reached. Then what we do is we wash off the excess ligand, we filter it or centrifuge it, and then do a few washings and make sure that there is no unbound ligand that is present. Now, if you measure the radioactivity, then that will give you an estimate of how much of drug is bound to the receptor in the cells or tissues. Okay. The extent of binding, therefore, can be measured by detecting the amount of radioactivity present in the cell and the amount of radioactivity that was removed, because by washing off we know how much of ligand has been recovered. So, based on this, we can define an equilibrium, that is ligand plus receptor going to a ligand receptor complex. Okay. And this equilibrium constant, much like what we defined earlier, is the concentration of the ligand multiplied by the concentration of the receptor divided by the concentration of the ligand receptor, or LR. Okay. And this is called as the dissociation binding constant. Now, the concentration of ligand we can find because we know how much has been added and how much has been recovered. The concentration of LR can be found by measuring the radioactivity of the bound ligand, and then of course, we need to correct for any background radiation, but we have no clue as to what the concentration of the receptor is. So, therefore, we have to use some mathematical manipulations to understand, or to either measure R or to remove R from this equation. So, we will now look at how this is achieved. So, the total number of receptors present must equal the number of receptors occupied by the ligand, that is concentration of LR, plus those that are unoccupied, that is R. So, we define a term known as R total, which is a sum of R and LR. Okay. What this means is that the number of receptors that is unoccupied by a ligand, that is R, is a difference between the total number of receptors and the ligand bound receptors. And as we know from our previous equation, Kd is defined as shown here. Now, doing a little bit of jugglery here, we can come up with this equation where the concentration of bound ligand divided by concentration of free ligand, that is LR divided by L, is R total minus LR, that term that I get from here, divided by Kd. Okay. And the total number of receptors present must equal, therefore, this sum of LR and R. Okay. So, we are still faced with the problem that Kd and R total cannot be measured directly. However, these terms can be determined by drawing a plot or a graph. Okay. So, what we do is we carry out a number of experiments where we use different concentrations of a known radioligand, and then we do what is known as a scattered plot. A scattered plot is nothing but a plot of Lr and L. Okay. And what we do is we plot the Lr divided by L versus Lr. Now, let us look at what happens when we do this. So, on the y axis, and this is the x axis, on the y axis is the concentration of the bound ligand, that is Lr, divided by the concentration of the free ligand. And as we have looked at earlier, using this radioactive experiment, we can find out the concentration of the bound ligand and the concentration of the free ligand. And if you plot that versus the concentration of the bound ligand alone, then you would expect to see a straight line, which is shown here, and the slope is a measure of Kd, and the intercept here gives you an idea of the total number of receptors that are present. Okay. So, using this plot, which is known as a scattered plot, one can determine R total as well as Kd. Now, if you repeat this experiment in the presence of the unlabeled drug, that is, this is the new molecule that we want to design, or we have designed or synthesized, and we want to study the efficacy, then if the drug competes with the radioligand for the receptor's binding site, it is called a displacer. Then what you would expect to see is a plot such as this, with x over here, and you would see a decrease in the concentration of Lr by L, because you are competing for the sites. The stronger the affinity of the drug, the more effectively it will compete for the binding sites, and the less radioactivity will be measured for Lr. Okay. So, what happens is that you will get a different line in the scattered plot. If the drug competes directly with the radiolabeled ligand for the same binding site, then the slope that we would see goes down. Okay. But then the intercept would remain the same, because as you increase the concentration of the natural ligand, at some point you are going to displace all of the inhibitor, and you are going to get a same concentration of R total. In other words, if the radioligand concentration is much higher, then the drug will bind to all of the receptors available. So, you should now keep in mind that this is very similar in a very similar perspective was applied to the enzyme inhibition as well. Now, agents that bind to the receptor at an allosteric binding site do not compete with the radioligand for the same binding site, and therefore, they cannot be displaced by high levels of radioligand. However, by binding to an allosteric site, they make the normal binding site unrecognizable for the radioligand, and so there are fewer receptors available. So, since there are fewer receptors available and the activity is happening in the same way, the slope of this remains unchanged. That means, if you have a slope with respect to the original ligand, the slope remains the same, but this concentration of Lr goes down. Okay. So, the data from these displacement experiments can be used to plot a different graph from which we can calculate some important parameters. So, what we do is we plot the percent of radiolabeled ligand bound versus the concentration of the new drug or the displacer. So, this, and we use a logarithmic term, and when we plot this, we get a plot as shown here. Now, this results in a sigmoidal curve, and it is termed a displacement curve or an inhibition curve. So, from this we can get what is known as an IC50. Okay. IC50 is nothing but the inhibitory concentration 50%, which means that it is a concentration of the compound that prevents 50% of the radioactive ligand from being bound. Okay. So, that value is shown here, and this value can be obtained by mathematical fitting to a curve. Okay. So, hormones and neurotransmitters, which we have looked at earlier, are very important endogenous molecules that are responsible for the regulation of a number of physiological effects. So, these molecules interact with a specific receptor in a tissue and they elicit a specific response. So, for example, we have looked at earlier the neurotransmitter acetylcholine mediates muscle relaxation through the central nervous system. Okay. So, if one were to measure the percentage of muscle contraction as a function of dose of acetylcholine, what you would find that at very low doses of acetylcholine, you would find that there is very minimal response, and as the dose of acetylcholine goes up, you would see that the function or muscle contraction goes up. Let me explain this plot a little bit more in detail. What we have done here is on the x axis, we have taken a negative log of concentration of acetylcholine. Okay. So, in other words, what this number 4 means is that it is 10 power minus 4 molar, okay. And here 10 means 10 power minus 10 molar. So, as we know, 10 power minus 10 is a number which is significantly smaller than 10 power minus 4. So, along the x axis, if I go from left to right, the concentration is going up. So, as the concentration goes up, the percentage muscle contraction is also going up. And you see that there is a region where there is not much of an effect, and then after it goes up, there is not much of an effect beyond a particular concentration. So, it is likely that at this top concentration, all the receptor sites are pretty much saturated, and therefore, no further muscle contraction is possible. So, now as most of the receptors become occupied, the probability of additional drug receptor interactions goes down, and the curve deviates from linearity, and you get a sigmoidal curve. Again, using mathematical fitting, we can obtain the very important terms that we can, for example, we can obtain an EC 50 or an efficacy concentration 50. Okay. Now, Kd, as we have looked at earlier, is the concentration of the test compound that gives half maximal binding. EC 50 is the effective concentration of the drug that elicits 50% of the total biological response. Okay. So, the biological response that we are looking at is muscle contraction. Kd we have looked at earlier as to how well it interacts with the receptor. Therefore, from this concentration response data or a dose response curve, we can measure EC 50 of the agonist or ligand. Okay. For an agonist, when we plot percentage of response versus concentration, we should get a curve that is similar, if not identical, to the natural ligand. So, here is a hypothetical plot of an agonist, and the original curve is shown here in red. So, as you can see, there is a very good overlap of the original natural ligand as well as the agonist. And so, if I determine the EC 50 here, it should be very similar in number. So, we will look at some examples during our problem sets. So, now let us look at what happens when we have a competitive antagonist. So, antagonist by itself will not show any response, and then when we add the natural ligand, here when there is no antagonist involved, you will see a curve like this, which is identical to what you would see earlier. Now, when you start adding a small amount of an antagonist X, much like what we would see with a competitive inhibitor, you would see that this X starts to compete with the receptor binding sites. Therefore, you would need a larger concentration of the natural ligand. Okay. Again, keep in mind, this is high concentration and this is low concentration. Okay. You would need a larger concentration of the natural ligand, which is acetylcholine, to see a response, and therefore, you would see a curve like this. And now, as we add more and more of the antagonist, you will find that the curve shifts towards the right. Keep in mind, since this is a competitive antagonist, at some concentration of acetylcholine, you would expect that you would still see maximal response. That means that the top part of the curve remains more or less unaltered, except that the curve shifts towards the right. Okay. So, we could also do this experiment in a different way. What we will do is, we can take a single dose of acetylcholine and vary the concentration of the drug. So, what we would see is that we will pretty much take a very high dose of acetylcholine, and then when the concentration of the ligand of the antagonist is very low, you would see no effect. That means, you will see 100% muscle contraction. Now, as you slowly increase the concentration of the antagonist, that means again here the concentration is low and this is high. As you increase the concentration of the antagonist, you will find that at some point you will start seeing a decrease in muscle contraction. And again, as you go forward and you go to very high concentration of the antagonist, you will find that there is no muscle relaxation, because all the receptor sites are now being occupied by the antagonist. From this curve, again by curve fitting, we can find out what the IC50 is. So, the IC50 value here is 10 power minus 7 molar. Like we looked at earlier, molar divided by 1000 goes to millimolar, then it goes to micromolar, and then it goes to nanomolar. Okay. So, 10 power minus 9 is nanomolar, 10 power minus 6 is micromolar, and so this compound is about between a micromolar and nanomolar efficacy. So, if we were to convert this, the value of this would be 100 nanomolar, which is not necessarily a bad number. Okay. So, for a non-competitive antagonist, what will happen is that, since it is we have already looked at the characteristics of a non-competitive antagonist, what would happen is that the maximal response will go down. So, again you see percentage of muscle contraction with varying doses of acetylcholine, you find a similar curve. Then when you add the non-competitive antagonist, the maximal dose will go down, that is maximal response will go down, because it is not competing for the binding site. Okay. So, two different binding sites may actually be involved. So, one where the non-competitive antagonist binds, and because it is an allosteric binding site, that the actual binding site of acetylcholine is going to be altered, and because of this alteration you will not see the same effect, you will not see the same effect. Okay. And so, when you have a non-competitive antagonist, what you will end up seeing is that the maximal dose of the, or maximal response, or in this case muscle contraction, will go down as you increase the concentration of the antagonist. But the IC50 here will remain the same, because what we are doing for IC50 is we are taking the maximum inhibitory concentration and then we are finding out 50% of that value, and so the IC50 will remain more or less the same. Okay. Now, when you see, let us take a hypothetical compound Y, and when you add it, and if you see a response such as this, where you see some increase in muscle contraction, then we would call this as a partial agonist. Okay. We have already looked at some of the aspects of a partial agonist before, and partial agonist is somewhere between basically an agonist as well as an antagonist. Now, finally, let us look at this concept of sensitization and desensitization. So, after the receptor has done its job, if there is a large concentration of the ligand, sometimes the receptor becomes insensitive to transmitting the signal. So, this process is called as desensitization. Desensitization can occur by a number of mechanisms. So, some of the mechanisms that are acting is by through phosphorylation of certain residues. So, let us look at what happens here. So, when let us say you have an agonist which is binding to a receptor site, and there is an induced fit, and you get the activity that we have is the ion channel that is open. But now, if the ion channel is open for too long, or if the agonist does not dissociate, then the cell responds in a way to prevent further ions from getting in by doing post translational modifications such as phosphorylation. And once this phosphorylation occurs, that induces a conformational change which closes the channel.